Thanks for joining me today, Ryan. I was hoping to get your thoughts on a few questions I had about the microbiome. So you've been a program officer at NIH for the past 8 years, and so I wanted to hear about how you've seen research around the microbiome change and develop during your time here.
It's changed over the last 5-7 years as there's literally been an explosion of research publications and countless scientific reports. And the focus has been actually looking at novel associations between various disease states such as diabetes, cancer, autoimmune disease, just to name a few, and the microbial population. So these associations and these research reports indicate that there are changes associated with these disease states in the microbial populations when compared to the reference database of the human microbiome and other consortia.
So what do you think is the connection between the microbiome and infectious disease?
First and foremost, the connection of the human microbiota and the host, the mammalian host, really can't be overstated. There's a number of research areas that have shown that there's an evolution of over millions of years between microbes and the host. And in fact, the microbes are absolutely essential for the development and proper functioning of the human immune system. The other area that I think is essential is that microbes form an innate barrier to invading microbes. And without these microbes, we become highly susceptible to infection. And the idea is that reducing these microbial populations or impacting them in some way makes us more susceptible to infection. That term is called colonization resistance. And mechanistically, it happens through a number of different areas, but nonetheless, both host immunity, development of host immune system, and the innate barrier, two areas that are central focus for NIAID research. And that's why I think that there's a significant connection between the microbiome and infectious disease.
So as a program officer, I feel like you're well suited to comment on the current state of microbiome research, as well as what are some of the big questions that researchers are actively trying to answer.
So I know it seems counterintuitive, but the reality is some of the big questions, we still don't know what constitutes a healthy microbiome. And the reason why this is particularly elusive is because there are many microbial compositions or populations that give rise to a similar function. So we can see a diverse sets of microbiomes, yet all of them be associated with health, if you will. And this even is compounded by geography. So there's, within various populations across, the globe, these compositions change. And so that's one of the big questions still is what's a healthy microbiome? And I think the answer to that's going to lie more in the area of functionality as opposed to composition.
Once we have a better understanding of some of these questions that you just highlighted, how will this affect public health?
From my perspective at NIAID, I feel there are two areas. One in the area of therapeutics, so so-called bugs as drugs, right? That's an often used term, and in the area of precision medicine, where you have individualized patient-level data, particularly at the genetic level, not necessarily the host genome, but the genomes of the collective microbiome on a person. So that area, precision medicine and therapeutics.
All right, thanks so much, Ryan, for enlightening us on this fascinating topic today. Appreciate it.
Absolutely. It was my pleasure, Ari.